Hi, my name is Danielle, and I am a student at the University of Wisconsin-La Crosse. My major areas of study are broadfield science, biology, and education. Basically, I am interested in teaching middle school or high school science as a career. I was asked to help with some scientific research about global climate change in Northern California by two of my professors, and I quickly agreed to go. Our research team took a plane from Wisconsin to California. You've probably heard about global climate change. Basically, scientific evidence shows that the climate of the Earth is changing, and many scientists have used this evidence to predict how the climate will continue to change over time. These changes will affect different parts of the world in various ways. For the grassland meadow in the Northern California coastal mountain range, where I helped with the research, climate models created by scientists predict that the area is going to get wetter. Our research is being done to test what would happen if our research site did get more rainfall over time. As you know, green plants need sunlight, carbon dioxide from the atmosphere, minerals, and water to grow. As plants, or primary producers, grow in size and greater abundance, we need to realize that we're not just talking about having more plants. We also need to look at which kinds of plants will be able to survive under the new conditions of more rainfall. How do we know how the plants in Northern California will respond to climate change and additional precipitation? Which plants can survive such changes the best? Since the amounts and kinds of plants will be affected by the additional rainfall in the area, organisms that feed on those plants, called herbivores, will probably be affected as well. Some herbivores will eat many kinds of plants no matter what's available, so they probably won't be affected as much by the extra rain. But there may be herbivores that only eat specific plants, and if the plant that it eats does not do well under these new, wetter conditions, then that herbivore will either have to adapt to new ways, or it will die out. This will also be true for the predators that eat the herbivores. At our grassland research site, plants, herbivores, and predators all make up a food web. See how climate change affects the interactions in an ecosystem? This research simulates what might happen to the interactions between organisms in a grassland meadow in Northern California when climate changes, if the area becomes wetter. To test the prediction of what might happen, the scientists that conducted this research set up 36 plots in a grassland meadow in Northern California. Some of the plots were control plots, which are plots that did not receive any water, so basically they experienced normal, current rainfall conditions. The other plots received extra water in a way that matches the climate change prediction models for Northern California. To add precipitation to this area in a way that matched the prediction models, the scientists added water to the plots through an irrigation system and sprinklers. From this picture, can you guess which plots are the watered ones? I went to help as part of this research team in the summer of 2009. We went to the Angelo Coast Range Reserve in Northern California. You can see it magnified on the map here. When we got to the reserve, we worked in one specific grassland area called South Meadow. Scientists were involved, including myself. You may think of people that look like this when you hear the word scientist, but you may be surprised because not all scientists wear lab coats, work in laboratories, and hold test tubes. Rather, scientists come in many shapes and forms. On this trip, the field scientists that worked in South Meadow were Dr. Suttle, and this is pretty much his experiment, and then people from UWL. Dr. Thompson, a professor and one of the people that helped set up the experiment, Dr. Gerber, also a professor, Jesse, a student studying biology, and me. You remember this picture, right? These are the plots that were set up for the experiment. What I did with Dr. Thompson, Dr. Gerber, and Jesse was help collect data from half of the plots so that the data could be studied and used to see how plants and organisms might interact as global climate change affects the area. It might be helpful for you to see what the plots look like at ground level. Can you see the orange caps in the grass? Those mark the outside edge of one circular plot. I know it looks elliptical here, but if you are standing right in the middle of it, it would be circular. This line might help you see the plot better. Within each giant circular plot, there were a number of smaller plots, or subplots, or mini-plots that we used for sampling. In this picture, the white frame marks one subplot within the larger plot that I was working in. We only used this white frame twice per plot, so we only took samples from 2 subplots per one big plot. All of us worked together to collect the plants in these subplots. To collect the plants, we used scissors to cut out every single plant at ground level within the square frame. Each frame is 1/4 meter on a side. When we were done, the subplot looked something like this. We then put all the plants that we cut into brown paper lunch bags. Each bag was labeled so we didn't forget which plot and subplot the sample came from. After we collected our samples from the plots, we began to sort the materials from each lunch bag. We separated the plants into piles of similar plant species. Each pile of a similar plant represented a separate functional group. Some of the functional groups we had included annual grasses that die every year, perennial grasses that come back every year, and then different kinds of forbs. We had to sort over 30 bags of plant material. After all of the plant samples were sorted into functional groups and placed in paper bags, we put the bags in a drying oven. This machine is just what it sounds like. It dries the water out of the plants by baking them at a specific temperature, just like an oven. All of our little paper bags were put inside a big brown paper bag to save space, as you can see in the picture here. We then measured the weight of each functional group of plants to determine their biomass. That's why this cutting, sorting, drying, and weighing process is called biomass sampling. With biomass sampling, we took a look at how much of each type of plant was present in each sampled control and watered plot. The kinds and amounts of plants varied among the different plots. For example, some plots had more annual grasses than others, so the weight of the annual grasses in those plots would be higher than in the other plots that didn't have as many annual grasses. With biomass sampling, we were able to collect some data about the amounts and kinds of plants present in each plot. We could also see if the plots responded to water treatments. If the plants grew more in response to additional water, they would accumulate more biomass and therefore weigh more. This data has been collected year to year and compared to see if any changes occurred as time passed. There are pros and cons to using any kind of experimental design, and in this experiment, biomass sampling is a useful method for tracking changes in plants over time. The only problem is that it requires that we temporarily destroy the subplots when we cut out all of the plants, as you saw earlier. So, each year, you have to sample from a new place in the larger plot. A less destructive method to see what types of plants were present in the plots is called point frame sampling. Dr. Thompson used a fancy contraption for this sampling method. You can see some wires with yellow flags at the top in this picture. Dr. Thompson would move each of the stakes a specific way, and at a chosen point, she would call out to me which plant was at that point. We did this many times in each plot to get an idea of which types of plants were present. I recorded the names of the plants with a specific shorthand code so that Dr. Thompson could easily input the data into her computer later on. Well, next, the data that we collected needs to be put into data sheets, and this is easiest to do on a computer. Once the information is put into data sheets, the data can be easily analyzed and compared from year to year. Good organization of the data is especially important since this experiment has been going on for almost 10 years. After everything is inputted and analyzed, conclusions can be made to try to explain what is happening out in South Meadow and why. To get even better data to support those conclusions, the experiment is continued year to year, and this whole process is repeated over and over again. What do you think the results of the experiment were? Remember, this experiment has been going on for many years, so think about what some of the long-term effects of extra rain might do to the organisms in each plot. Try to think about interactions between plants as well as insects. You can work like a scientist too and test your hypotheses in your own schoolyard.